Duchenne muscular dystrophy, or also known as DMD, is an X-linked recessive genetic mutation. On the X chromosome, there is a small deletion of DNA that results in the lack of production of an important protein called dystrophin. Dystrophin is a part of an important complex found at the membrane of muscles that stabilizes the membrane. Without this dystrophin, this complex would not be able to properly bind to the membrane, and the membrane would be much more unstable and prone to injury. In DMD patients, contractions induce microlesions in the membrane, which lead to cellular dysfunction and degradation. One mechanism of action by which DMD affects tissue is through these high regeneration rates. Initially, this high cellular degeneration results in a high regeneration rate. However, muscular stem cells have limited regeneration capacity that is reached at a relatively young age in patients with DMD, eventually leading to a net muscular degeneration. The provided muscle biopsy shows a lower cell proportion between affected and normal cells of mature tissue compared to younger tissue, pointing to the idea that the stem cells are losing their regenerative capacity. This net muscular degeneration, along with other factors, causes the destruction of muscles in the body, including important ones like the diaphragm and heart. DMD is the most common form of muscular dystrophy, affecting 1 out of every 3,500 newborn boys every year. Because it is an X-linked recessive mutation, DMD is almost exclusively found in boys. Often manifesting from the ages of 2 to 7, DMD causes a steady decrease in muscle strength and stamina because of the worsening muscular dystrophy as they age. This decline results in the eventual non-ambulation, which is the inability to walk of boys around 12, and most commonly, heart and lung failure in their teenage years to early 20s. There are no existing current therapeutic drugs that make a significant difference to the DMD condition. DMD patients have full control of eye movement, which are actions controlled by muscles called extraocular muscles, or EOM. EOM are arranged around the eyeball and contract and relax together to control eye movements. If DMD were to affect EOM, DMD patients would not be able to control the movement of their eyes towards the later stage of the disease, which is contrary to reality. Additionally, they also express the exact same dystrophin complex that is found at the membrane of muscles, which begs the question, if muscle degeneration occurs because of contraction-induced injuries, then why are EOM muscles spared in the muscular dystrophy that is seen in the rest of the body? Currently, the most promising approach is the study of EOM stem cells. EOM has a population of stem cells that are activated and proliferative, even through aging. Stem cells are cells in humans that haven't differentiated yet and can be used to develop new cells or more stem cells. Stem cells in the context of DMD are crucial in restoring the frequently destroyed muscle cells. The most significant and exciting experiment took MDX mice, which are the mice DMD model, and put EOM stem cells into skeletal muscle. They found that EOM began to differentiate into skeletal-like muscle cells, expressing muscle-specific genes such as PAX7. This proves that EOM stem cells can differentiate into satellite cells, which are muscle stem cells, and have a high probability of continuing to differentiate into fully grown, healthy skeletal muscle. EOM and limb stem cells from MDX mice were taken and transplanted into the abdominal muscle of another MDX mouse. The results were impressive. The EOM stem cells were shown to retain higher proliferation performance and adaptation compared to that of the limb stem cells. Successful transplantation of the extraocular stem cells suggests that possible clinical trials of these transplantation of these stem cells will be successful. So where can we go from here? From a research perspective, one can read on current literature and perform experiments to expand on that knowledge. From an individual/parent/advocate perspective, one can vote on policies to increase funding for stem cell research or have conversations with local politicians and communities about potential benefits of focusing on extraocular muscle in DMD patients. We know DMD as a whole is a very wide and multi-approach disease that can have a variety of therapeutic drugs, but perhaps looking at DMD from a fresh outlook can point to a new possible therapeutic drugs that can impede or reverse the effects of dystrophy on in DMD patients, helping boys lead longer and fuller lives.